So it's really exciting for me to be able to talk to Walter Isaacson about his book, The Code Breaker, which is something very much in my wheelhouse as the editor of Science and a nucleic acid chemist. So I have to kind of repress my younger self who can't believe I'm actually talking to America's top biographer about nucleic acids, but we'll try to keep it under control here.
And I'll try to get David McCullough here to be the top biographer for you.
Walter Isaacson is well known to you all, I'm sure, but he grew up in New Orleans, where he lives now. He went to Harvard for undergrad. He was a Rhodes Scholar. He had leadership roles at Time, CNN, and the Aspen Institute. He has written important biographies about Steve Jobs and Ben Franklin and Albert Einstein, and as we're here to talk about today, Jennifer Doudna, someone I've known since we were both postdocs at Yale 30 years ago. So that's super exciting to be talking about her. It's such a great thing for science that she's the focus of Walter's latest book. So Walter, I think we ought to start kind of at the beginning. I mean, one of the things that I love about the book is you open it. There's a little preamble about Jennifer finding out, and Jennifer, Dr. Doudna, Nobel laureate. Okay, but I've known her for 30 years, so I call her.
Wait, I'm going to interrupt.
Yeah.
What was she like in grad school?
Oh, she was, we all knew this was all going to happen.
You knew it?
Yeah.
Did you say Nobel Prize when you first met her?
Yes. Yeah, because RNA structure was going to be the future. We knew that.
That's the theme of everything these days. Gets us our vaccine.
Yes, absolutely. So, there's a little preamble about Jennifer finding out about COVID, but then, it goes to where every scientist would love for the book to start, the central dogma, nucleic acids. So how did you think
You're
about...
not doing well at selling this book. The central dogma of nucleic acids. This is the problem of having the world's greatest science editor.
So how did you decide how to open the book in order to kind of bring people to where they can understand what CRISPR is?
Well, it was interesting. I'd about 2/3 of the way through reporting the book, and it was the first time I had done somebody who was still in mid-career. So I'm thinking, how are we going to get, a climax to this book? And then 2 or 3 things happened. One, COVID strikes, and the structure of molecules becomes quite important. Number 2, she wins a Nobel Prize. And so I just delayed the book for a while and said, we're going to tie it all in to the coronavirus pandemic.
Right. And that worked out magnificently. But I guess for the audience, bring them up to speed on what CRISPR really is and, the...
CRISPR is simply a very easy-to-use tool, very reprogrammable, so easy that I did it in a day and a half to edit your genes. Now, you got 3 billion base pairs of letters in your DNA. And what CRISPR can do is say, all right, this one's wrong. It's causing you to have sickle cell. Or this one, if we edit out all these genes, you're going to be a foot taller. That's a little bit further down the road. Or, we could do all sorts of edits, especially genetic diseases like sickle cell, muscular dystrophy, cystic fibrosis, and then diseases that are predisposed by genetics, including maybe schizophrenia or manic, psychological diseases. So it allows you to make edits of a gene. Why is this a big deal? Species have been around for approximately 3 billion years. For the first time, one species, happens to be ours, has figured out how to hack its own evolution and to design its own children, change its own genetic structure, fix its own genetic problems. Specifically, what CRISPR is something bacteria have been doing for about 1 billion years. And it ties into the coronavirus pandemic because viruses attack bacteria. Bacteria have got the same problem we do. They keep getting hit by viruses. And what a bacteria does is it takes a snapshot of the genetic material of the virus that's attacking them and makes it into a mugshot that they weave into their own genome. And it's done in these clustered, regularly interspaced palindromic repeats. Well, that's what CRISPR stands for. Now, the reason it stands for that is there was a wonderful Spanish scientist named Francisco Mojica who was noticing these repeats, and everybody was trying to come up with a clever name. So he comes up with the clever name, which is CRISPR, and then he has to reverse engineer a whole bunch of words to make it into CRISPR. So you don't have to remember all those words. You just have to, CRISPR is this way that bacteria chop up genetic material. And what Jennifer Doudna and her partner Emmanuel Charpentier do that wins them the Nobel Prize is say, we can re-engineer this. We're as smart as bacteria. If they can do it, we can reprogram it. So if they're attacking this type of genetic material, why don't we have it cut the sickle cell mutation or the muscular dystrophy mutation? So they repurposed a system that bacteria have known as CRISPR to allow humans to edit their own genes. And it's a big deal.
Big deal, yes.
And it's a big deal, I'm sorry to go on, but like I looked at the physics revolution with the Einstein book. That was a big deal. We split the atom, we did GPS, we went to space. And then, of course, I looked at the digital revolution with Steve Jobs and others, and that was a big deal. And we got iPhones. This is, I won't use Joe Biden F word, but it is a big deal,
yeah.
Which is editing our own DNA makes molecules the next microchip. We'll be able to reprogram our molecules the way we do our microchips.
Right, and it's really exciting what we might be able to do. So you've written about a lot of old men, most of whom aren't here anymore.
Yeah.
Steve Jobs you got to interview, but he had passed away when the book came out. Now you're writing about someone who is still very much in the middle of her career, and she keeps sending me a lot of great papers, so she may win another Nobel Prize for all we know. What do you, how was that different?
Well, it was different because this is not so much a history book as a journey of discovery in which I feel I'm doing it, and we all are doing. Reader is doing it with her.
Yeah.
In other words, when you start the book, we don't know how we're going to get out of, you know. And so by being with her in her lab, you get to tell a story that's a narrative in real time, as opposed to, and then Dr. Franklin sailed off to Paris to do the peace accords or something. Or Henry Kissinger sailed off to Paris to do the peace accords, whatever those people going off to Paris to do peace accords. And so it becomes more of an adventure story that is told in parts. And for those of you, or many of you are writers in this room, you'll see a delicate, tricky thing I had to do is I sometimes intentionally go into the present tense. You know, she walks into a room instead of she walked into a room, as things are happening as we're reporting it.
Yeah, I saw an interview you did with Doris Kearns Goodwin where the two of you were talking about your process. And one of the things that you talked about was the chronology that, you know, you use as kind of your touchstone as you're doing these books. Tell us a little bit about that process.
As I mentioned earlier, I had a mentor, Walker Percy, the novelist, when I was growing up in Louisiana. And he said there are two types of people come out of Louisiana, preachers and storytellers. He said, for heaven's sake, be a storyteller. The world has too many preachers. And then when I sort of sat at his feet to understand that, he says, a story is when you tell things in chronological order. You make it so one thing leads to another. And I said, well, that's pretty amazing. He said, no, that's the way the Bible did it. You know, best lead sentence ever. In the beginning, comma. You know we're going to get a narrative. Oddly enough, many history writers and even many biographers don't make things into a narrative. They don't make it so that at graduate school in Yale, when you all are studying with, well, I guess Jack Szostak at Harvard, then Tom Cech, and Seitz was probably the person at Yale you were doing.
I was studying photosynthesis.
Oh, okay. Well,
yeah.
But whatever you learn there is going to make a difference to what you are the following year when you get asked the question, how did life begin on the planet? And you say, okay, that builds on. So you want to do everything in chronological order. I had an editor who died a couple of years ago, but she had edited my other book. She said, scrawl in the margins anytime you got something out. She'd say actigta, which meant all things in good time, meaning don't get ahead of the narrative, don't get behind it, just keep it straight narrative. Now that seems boring, and what you have to prevent with that, and this is what Doris does so well. I mean, the greatest of narratives, say, is take the bully pulpit, where she has Teddy Roosevelt barnstorming the country by train of a fair deal for the American people or whatever. It's got to each build up into a climax. What you want to avoid is it just being one damn thing after another. It has to have a narrative arc. And so that's the process. Speaking of Doris, so when I was a student, when I was a young undergraduate, there was a great course at my college called the American Presidency, set by a wonderful guy named Dick Neustadt. So I signed up for it, and Dick Neustadt decided to fall in love with somebody in England and left for a year. And so this very young...
have the funniest way of doing that.
Yeah. And so he had this young graduate teaching assistant named Doris Kearns, and she takes over the course. So she becomes my teacher, and she hasn't even written her dissertation yet. And a couple years later, she writes her dissertation, which is Lyndon Johnson, the American Dream. Harvard denies her tenure because it's a biography,
right.
A great man theory, biography, narrative biography, and that's considered beneath the dignity of the academic world. You're supposed to write about great forces of history or Village in the Vaucluse or whatever everybody else was writing about at that period. So she's denied tenure, and that sends a bit of a shockwave through the academy. And nobody writes biography if you're a professor then, because this is a pretty famous thing when she gets denied. This opens the door for non-academics to take over the field of narrative biography. And so you have Bob Caro, you have David McCullough, you have a whole generation of people older than
Taylor
me.
Branch.
Yeah, Taylor Branch, we can name a whole
Yeah.
bunch, who are partly journalists, or in Bob Caro's case, and they take the field of narrative biography and do it. And then, when I was a journalist, it was like, okay, this is a field that other people aren't plowing. Mm-hmm. And, John Meacham, myself, Evan Thomas, a lot of my friends, we tried to be the successor generation to the McCullough generation.
Right. And that brings me to an interesting point about the book, because I think one of the things that we really long for in science that this does is to bring this narrative and this human drama to discoveries, especially those that are happening right now. I mean, we don't have a lot of TV shows about scientists. Most people think that we're just sitting in some room staring at a screen being completely objective and unemotional, but yet we have every aspect of human drama that you have in any other human endeavor. And I think the fact that we haven't explained that is a big part of why people are having a hard time understanding what we're saying
the whole
about the COVID,
anti-science and resistance to science problem is mainly the result of yahoos who are resisting science. But there's a 10% blame that I think goes to the scientists, not including you, because what you're doing, especially with Science Magazine, is incredibly good. I mean, it's both reliable and readable, and you're popularizing as well as drilling deep into peer-reviewed science.
Unfortunately, Ben Shapiro's posts get a lot more traction than ours do on Facebook.
And so what we need to do is demystify science and make it seem like a human endeavor so that people can relate to it. They're not intimidated by it. And one of the purposes of the book is to describe what actually happens in a lab. What does a scientist actually do? What is Jennifer Doudna doing to make this discovery and stuff? So I try to make the lab into a journey of discovery, just as if you were going into space or traveling to settle Missouri in the beginning of McCullough's Truman biography. Whatever it is, it's a journey of adventure. And after I wrote about Ben Franklin, I was surprised how amazingly important he was as a scientist. We think of him as a doddering dude flying a kite in the rain, but those electricity experiments are the most important experiments of that era, and the single fluid theory of electricity becomes the most important theory. And he or Jefferson would have thought you were a Philistine if you didn't believe in science. And it's the whole point of the Enlightenment of our Constitution is the empirical balancing of checks and balances and evidence gets us closer to the truth. So what happens? When I was growing up, there was the C.P. Snow Great Divide between people who love scientists, people who love the humanities. And partly it was Einstein's fault, because Einstein comes along and suddenly you have to be an Einstein to understand things. You know, relativity is not the same as Newton, checks and force and mass. And so I said, I want to demystify science. And the best way to do it is to turn Einstein's journey of discovery into a human thing you can relate to. And so to me, we need more of that. There are great people, scientists doing. I love Brian Greene. He's in the tradition of Asimov or of the famous Sagan, Carl Sagan. But I wish we had more popularizers of science. Unfortunately, I'm sorry to go on to this, just like the academics look down upon Doris Kearns by being a biographer, scientists sometimes look down upon popularizers.
Not sometimes. It's a major problem.
It's a major problem for the scientists because if you don't have popularizers, people are going to be not appreciative of what you do.
Yeah, scientists look down on that and then at the same time complain that nobody understands what we're doing.
Bingo.
Yeah, right. Yeah. As you were learning a lot about how science is done now, the competition between the Berkeley group and the group at MIT and them sending us papers and us accelerating it because there was a competing paper at Nature and a lot of this back.
He plays a big role in this story. They're all trying to get into Science magazine. And when they can't, they get into Nature.
That's right. That's how we like it. Yeah. Science is in the index 42 times and Nature only 23, in case you...
But who was counting?
Yeah, not that we're competitive or anything. So what surprised you, or did you not kind of know about the way all this is done?
Well, it first surprised me how competitive they could be, especially you're talking about the bitter, still in court competition between the Doudna-Charpentier women and the Eric Lander Fong Jiang Broad Institute, MIT, Harvard crowd. And what surprised me shouldn't have, because, anybody in any field here who thinks about it thinks, okay, sometimes I cooperate and sometimes I'm competitive. Whether I'm on the softball field or whether, we're journalists and trying to do things, it's like, all right, I'm competing. And you have to get that balance right. I think three things tip the balance badly or towards the bad side when it comes to competition versus cooperation, collaboration, which you want the right balance of. First was patents. And this happens, there's a large section in the book of the patent battles. And suddenly science becomes business and commercialized, and if you discovered CRISPR, you want to make sure you get the patent. So that's the thing. Another thing, and you may disagree, I want to hear your opinion on this, I think prizes are a problem. The Nobel can only be given to 3. The breakthrough is given to 1 or 2 or whatever. And so you're trying not to share with your friends at the Broad because you're competing against them for the Nobel Prize. And the third involves you directly is prizes, patents, and priority of publication. And that's the race. And so when Jennifer hears the footsteps of people, doing CRISPR-Cas9, which was a great June 2012 paper that you published, I'm sure, well, I know, or at least it's in the book, I hope it's right. She is pressing the editors of your magazine to get the damn thing in fast because, other people might publish first.
Yeah, and we did, and it was good training because we published all the COVID papers very fast, and we learned to...
And one of the things that happens with COVID, and I will turn this to you to explain, is it does change that nature of peer reviewing and waiting 3 months to, and you see both bioarchive, meaning open source servers, and then science hitting the seal of approval on it. But it also caused Jennifer and her rival Fong Jiang to quit competing so much because when they were applying these discoveries to detection technologies for the coronavirus, they were publishing them every evening openly and not asserting intellectual property rights.
Yeah, I mean, I think the excellent story about COVID that underlies all that is that's similar to Doris Kearns Goodwin not getting tenure, which, thanks for reminding me why I'm glad I'm not a provost anymore, is that the people who really came up with the idea of the mRNA vaccines, Katalin Kariko and Drew Weissman, Kariko did not get her to get tenure at Penn. And
yeah, I mean, Penn is deeply embarrassed. Yeah. They've had Donald Trump, who they did get to be president, and Katalin Kariko, who they didn't give tenure to. Katalin Kariko, for those of you who don't know, is a great story.
Yeah.
But she's the person who everybody knew, since you started with the central dogma of biology, the central dogma of biology is that messenger RNA tell our cells what proteins to build, basically, and they get the info from our DNA. That's easy enough, but you have to figure out, if you're going to make a vaccine, how do you actually make it work in a human cell? How do you get it into the human cell? And that's what this Hungarian student at Penn figures out, and then they deny her tenure.
Yeah, well, she's going to get a Nobel Prize, I'm pretty sure. So let's setting aside whether it's a bad thing to give Nobel Prizes or not. Okay, so we have some time left, and we want to get some questions from the audience, if there are any. If anybody wants to come to the, sure, go right to the microphone there. No, we got him first. I'll come to you next. Yeah, go ahead.
Yeah, thank you both. Dr. Edward de Bono passed away a few weeks ago, and The Economist dedicated a page to his obituary. He wrote 70 books about creativity and thinking, coined lateral thinking. And what drove him in his early medical career out of Oxford was the idea that you could reprogram the brain. So CRISPR, a person a foot taller, CRISPR, a better brain?
Yeah, obviously the brain is the most complicated of our systems, and there are things that are genetically determined in terms of synapses or hormonal, and those things can be tweaked with CRISPR. As we go down the road of what are we going to tweak and what are we not going to tweak, in the book, I get to the brain last because that seems like, wait, we're not ready for that. Obviously, I think we should start with single gene mutations that cause really bad disorders, such as sickle cell or muscular dystrophy, cystic fibrosis, etc. And then you can go down a slope that can be slippery if you don't go down cautiously to say, and here we would do this, like people who are born very short, maybe fix that, but maybe we don't do enhancements. If you're going to be born, 5'9", we don't give you an extra 8 inches, but if you're going to be born 4'9", you do. Anyway, I'm droning on, but there's a whole section in the book, and the last is, do we deal with depression? Do we deal with schizophrenia? Do we deal with wanting more synapses? Creating more synapses for more memory would be rather easy. Well, maybe we do that to prevent Alzheimer's, but do we do it to make super kids? And I think there's moral issues that come along when we start designing things, so we got to go slow.
Yeah, there's a great section in the book about that. Yeah.
So I enjoyed your book very much.
Thank you, sir.
In your book, you focus on trying to slow down the use of CRISPR in humans. And I want to bring up a slightly different perspective, and that is I've worked with a couple of genetic endocrine cancer syndromes for the last 40 years, and about 25 years ago, we acquired the ability to do pre-implantation screening and in vitro fertilization and to eliminate the genetic defect from the germline. In the last 20 years, I've had exactly one couple, even though I bring it up in my interviews with these patients. Mm-hmm. And I'm not sure, based on that experience, that CRISPR is going to be used any more than a technology we already have? And I wonder if your thoughts.
It's a good question, and I think everybody in this room has as good a thoughts as I do, because you can look inside yourself and say, all right, suppose now, real quickly, as you know, pre-implantation diagnosis means creating a lot of fertilized eggs and testing them, and we actually, went through certain fertility things. It's not as easy as it looks for a couple sometimes to say, okay, I've got all these embryos, let me pick one or the other. So CRISPR allows you, as it happened in China, to just say, let me edit it. So it becomes quicker. The question is, and I do the thought experiment in the book, as you know, is suppose you're at a clinic, and it's almost like a genetic supermarket. And you're going to have a baby, and you've seen the movie Gattaca, and you've read Brave New World, and they give you a list of things. Do you want to make sure that your child doesn't have Huntington's, doesn't have you go, of course, of course, of course. Do you want your child to be above average? Well, of course. Above average memory. Do you want to have sickle cell? No. Do you want to have extra oxygen in the blood? Meaning, instead of sickled cells, cells that carry more oxygen. So he or she will be clearly a super athlete if you do that. And you're sort of getting a little queasy, but you probably check yes, And then we all have to figure out how far we would go if it were easy enough just to choose attributes for a designer baby. And I think we have to start by doing that thought experiment. And maybe you're right that only 20% of the people will want to design the characteristics of their baby. But each person in here can do the mental calculation. You don't have to reveal it to the person next to you, which is how likely would it be if it's your grandchild having a kid and you're with them that you'd say, hey, I'll pay for that premium option. I'll make the kid taller for you.
Yeah. Thank you. Yes.
Before the event, I approached Walter at the book tent when he was signing the book to complain virtually hysterically about the absence over the past 2 years of animation.
Yeah, Deborah and you have both hit me on animation. I write words on a page.
I'm not talking to you, bro.
Oh, well, he does it very well in
We
science.
have a lot of animations.
And if I may
sciencemag.org.
continue, Walter suggested I come see you immediately because Science Magazine has produced or has made available the very animations that I'm looking for the past 2 years. So it's great to know that they exist on the planet, either in this country or other countries, or there's a collective pool of animation that will show us the virology, the biology, the immunology of COVID and everything else, given that there are millions of Americans and people all over the planet who have the equivalent of BS and BA education. I was in college in 1966 when Watson and Crick had their breakthrough, and we read the book immediately, and ever since then I've been fascinated by DNA. So please make the animations available where everyone can see them.
Yes, well, sciencemag.org, and there's lots of stuff there.
Everybody has gone to this already.
Yeah.
They have huge
but,
animations.
you bring up a really important point because, as Walter was alluding to earlier, science has done just a terrible job of inviting people into what we do. And so even our website, which is really mainly written for other scientists, is certainly a place that has a lot on it and some of it pretty accessible. But there are other things too. I mean, I happen to be a big fan of the Khan Academy videos, and I usually recommend people start there because they really do a nice job of.
And Carl
starting at
Zimmer
the
in the New York
beginning.
Times has amazing animations on the
Superb,
virus
yeah.
and on the vaccine.
Yeah. Thank you.
Thank you. This is wonderful. Could you discuss other countries' policies and which countries you feel are more competitive to us because of the various restrictions and difficulties you've already pointed out, such as getting tenure, patents, lack of cooperation, and where America sort of fits in the bigger picture?
Mm-hmm. I'll do it on genetic editing, which I'll focus on genetic editing, which is when Jennifer Doudna made this discovery of how to edit genes easily, she had a nightmare. And the nightmare was somebody wanted to figure out the technology with her. She goes into the room, and it's Adolf Hitler. And so she decides to start gathering scientists from around the world to say, what restrictions, what rules of the road are we going to put on this? The places in which it is furthest along is, not surprisingly, U.S. and China. And then Germany, quite a bit, is doing a lot. So far, there have been an international consortium of scientists, to say, here's the guidelines we'd like to have. And even China, where it was done to create designer babies by a scientist who wasn't fully authorized to do it, but just did it, who had been to one of Jennifer Doudna's seminars, went back to China, and made designer babies, he's now in jail because China and the U.S. are working very closely together to say, let's not get ahead of this before we figure out the rules of the road. The one place I worry about is Russia, because Putin has pushed this. Putin has given speeches saying, we're going to create super soldiers, we're going to create, and Russia is less likely to be involved in an international consortium.
Yeah, and I think that science always in America has been funded based on international competition. The reason we have a federally funded science organization in the United States is because of the end of World War II. Then we had a Cold War of Russia. Now if you listen to the people who go to authorize money from Congress for science, they're talking about competing with China. But at the same time, we need to collaborate with China because they got some great scientists over there. And so American science has always walked this line between getting the money because we're competing, but wanting to have international collaboration so that we can get the best discoveries. And that's an ongoing...
And Eric Landers, we should say, who is very much in the book, is now the chief science advisor to the president.
Yes, and Eric, there's a lot of great stuff in the book about Eric. And he's got a big job because he has to walk these two lines. He's the OSTP director, which is Office of Science and Technology Policy. He's the science advisor to the president, and thankfully Joe Biden made that a cabinet level position, and Eric was confirmed, which made Walter and me very happy. Yeah.
Quick question. I think a lot of people in this audience will be interested in this, which is where we are as far as reversing the aging process. And that's a general question, but I'm sure you can get more specific. But something that you said earlier, which is, you can deal with diseases and whatever, but the brain is further on. And I wanted to talk about that a little bit because I don't think anybody here wants to be an empty vessel. So could you address that?
Yeah. Well, there are two components of aging process research, and maybe Holden can fill in more. One is the sort of the telomere thing where you try to do it in a broad brush way by slowing the shortening of telomeres. The other is, what is it about aging that you particularly want to stop? And some are simple, like osteoporosis. Well, okay, that can maybe be genetically fixed. And the more complicated one, which you just referred to, is memory. And that spans everything from Alzheimer's research, which has really not been as successful as we would have hoped, to genetic engineering to create more memory in the brain, which could happen. And so it doesn't necessarily lengthen life, but it lengthens cognitive skills, which is, to me, and I think you kind of implied that, the more important thing. And I think within 5 to 10 years, we will be able to use genetic engineering to deal with memory and to increase memory, and something probably dangerous, which is to have kids who have much better memory. I was just reading a book, Kathy will remember, we went to the book thing, Joel Klein had it, of a person who's written a book saying how important it is that our memories aren't great, that we couldn't do narratives, we couldn't do storytelling, it's a problem if your memory is too good. But I think we'll be able to enhance memory and test that theory.
Thanks. Is there a non-state actor or terrorist side to
Yeah.
CRISPR technology.
Yeah, there's a chapter in the book called Anti-CRISPR in which DARPA, the Defense Department's research agency, is funding Jennifer's lab as well as the MIT lab to create anti-CRISPRs, which is what it sounds like, anti-ballistic missiles, try to stop missiles, how to... and in fact, around here was a big deal because Kevin Esvelt and others at MIT, meaning not my crowd at Berkeley, were thinking of gene-drive mosquitoes in order to, I think, stop Lyme disease or whatever it was.
Probably malaria. Yeah.
Malaria, I don't know. Is malaria a big problem on Martha's Vineyard or not?
Yeah.
But, you know, well, that's fine. But I remember being a young kid when they came up with ways to get rid of mosquitoes and DDT, and all of a sudden in Louisiana we didn't have any more pelicans. So you have unintended consequences when you unleash things like this onto the world. So the Defense Department wants to know how to reverse some of these things. Now, there are two types of actors you have to worry about. There's the biohackers, some of whom in my book, and I find quite amusing, you know, and they're doing things, biohacking CRISPR so they can have better muscles by regulating myostatin. And I think that's harmless, but clearly if you had a biohacker who was evil, you could create really bad things. And so that's just the non-state actor. And then you do have to worry about state actors, and I won't get into the controversy here because I'm still confused by it. But even with the SARS-CoV-2, you know, to what extent were there gain-of-function experiments that may have accidentally led to some of these things? Well, that's what this is, a book about gain-of-function experiments.
Yep. On complicated organisms. Yeah. This will be the last question.
The Lion King.
Yeah.
Yeah. I'm kind of wondering about the genetic modification used on coral reefs and how they can combat against bleaching events and what effect they might have on marine life later down the road, if you know anything about that.
I don't. I don't know if we need another last question. I'm so sorry. I should. I don't even know whether coral reefs have genetic material, do they?
Oh, sure. They're alive.
I just have to end by proving how little I know.
Yeah.
So
Yeah.
I'm very sorry.
There's only one I know of in Hawaii.
Yeah.
There's only like one experiment I know of in Hawaii, so
Yeah.
it's like it might be early on with that.
Well, I hope you write a piece for it. Send it to me because I know the editor of Science Magazine.
Well, I happen to be a freelancer, so.
We're
How
always
much
looking
do you
for
pay
great
per
stuff.
word?
It's
Actually, they pay per
scandalously low.
footnote, right?
Yeah, there you go. Yeah, so Walter, thanks so
Thanks.
much for doing this. Thank you all.